Hello everyone, welcome back. In this video, we'll be trying our best to finish the unit revision, and in this video, we will be using physics and math tutor notes. But as usual, we used international UK AQA notes. But in this video, we'll be using the AQA notes as PMT has very nice key points for each chapter that we will be trying our best to use and finish by today. So in this video, we will be focusing on the biological molecules. And if we had time to take another thing, we'll be writing as well in the, in the title of the book, of the video. So let's get started. Starting with the monomers and the polymers, you need to know that a monomer are small units which are the components of larger molecules. For example, it includes monosaccharides such as glucose, amino acids, and nucleotides. Polymers are molecules made from many monomers joined together. So monomers are joined by a chemical bond in a condensation reaction whereby a water molecule is eliminated, while hydrolysis is the opposite of condensation reaction and is when water is added to break a chemical bond between two molecules. First of all, the biological molecules are divided into many types. First is the carbohydrates, where carbohydrates are molecules which consist only of carbon, hydrogen, and oxygen, and they are long chains of sugar units called saccharides. A single monomer is called a monosaccharide, with a pair of monomer being called a disaccharide. So a disaccharide are two monosaccharides, as di is two. Combining many monosaccharides result in the formation of a polysaccharide, and they are all joined together with a glycosidic bond formed in a condensation reaction. Monosaccharides such as glucose. Glucose is a monosaccharide containing 6 carbon atoms in each molecule, and it is the main substrate for respiration, and therefore of great importance. It has 2 isomers, alpha and beta glucose, with structure being on the right. Common monosaccharides include glucose, galactose, and fructose. These are typically sweet-tasting, soluble substances, and which have the general formula CH2ON, where N can be any number from 3 to 7. Alpha glucose is drawn like that: H is up, OH is down, beta OH is up, H is down. Disaccharides are 2 monosaccharides that are joined together in a condensation reaction to form a disaccharide. In this process, a molecule of water produced. The diagram below shows the formation of 1,4-glycosidic bond between 2 alpha glucose molecules in order to form a molecule of maltose. Maltose is formed by glucose and glucose. Maltose, the letter M stands for main, so the main monosaccharide, glucose and glucose, so that's they form maltose. Why 1,4? Because it's between carbon 1 and carbon 4. We have also sucrose, which is formed by glucose and fructose. You can see sucrose, fructose, they sound together like each other. And also lactose is by glucose and galactose. If you remove the GA from galactose, it's still lactose. So that's an easy way to remember it. So maltose is formed by a condensation reaction of 2 glucose molecules. Sucrose, glucose, and fructose, lactose, glucose, and galactose all has at least 1 glucose. Now we go to the polysaccharide, which basically are formed polysaccharides are formed from many glucose units joined together and include glycogen and starch and cellulose. Glycogen and starch are both formed by condensation reaction of alpha glucose, while cellulose are formed by beta glucose. Glycogen is the main energy storage molecule in animals and is formed from many molecules of alpha glucose joined together by 1,4 and 1,6 glycosidic bonds. We're going to talk about it later, 1,4 and 1,6 thing. It has a large number of side branches, meaning that energy can be released quickly as enzymes can act simultaneously on branches, meaning that energy can be on these branches. Moreover, it's relatively large but compact molecules, thus maximizing the amount of energy it can store. Finally, being soluble means it will not affect the water potential of cells and cannot diffuse out of cells. This part is very important. Again, what is glycogen? Glycogen is the main energy storage molecule in animals and is formed from many molecules of alpha glucose joined together by 1,4 and 1,6 glycosidic bonds. So, glycogen is a polysaccharide formed by alpha glucose joined together by 1,4 and 1,6 glycosidic bonds, and it's the main energy storage molecule in animals. For your information, which is very important, it has a large number of side branches, which means it has energy can be released quickly as the enzymes can act simultaneously on these branches. Moreover, it's relatively large but compact molecule. Why is that important? It maximizes the amount of energy it can store. Finally, it's insoluble, which means it will not affect the water potential of cells and cannot diffuse out of cells. Now we go to the starch. Starch stores energy in plants and is a mixture of two polysaccharides called amylose and amylopectin. Amylose is an unbranched chain of glucose molecules joined by 1,4 glycosidic bonds, and as a result, amylose is coiled and thus a very compact molecule storing a lot of energy. Amylopectin is branched and is made up of glucose molecules joined by 1,4 and 1,6 glycosidic bonds. As you recognize over here, glycogen has 1,4, 1,6, amylopectin has 1,4, 1,6, amylose has 1,4 only. Glycogen is branched, amylopectin is branched, amylose is not branched, so 1,6 form branching. Okay, amylopectin, due to the presence of many side branches, these can be acted upon simultaneously by many enzymes and thus broken down to release its energy. So it has two pros. So some key properties of starch that make it suitable are that it's insoluble, so will not affect cell water potential. It's compact, so a lot of energy can be stored in a small space, and when it's hydrolyzed, the released alpha glucose can be transported easily. Let's wait for the other and complete after it. Let's get back. Okay, so cellulose is also a component in the plant, but it's a component of cell walls, and it's composed of long, unbranched chains of beta glucose, which are joined by glycosidic bonds. The microfibrils are strong threads which are made of long cellulose chains running parallel to one another that are joined together by hydrogen bonds, forming strong cross linkages. Cellulose is important in stopping the cell wall from bursting under osmotic pressure, which this is because it exerts inward pressure that stops the influx of water, which means that the cells stay turgid and rigid, helping to maximize the surface area of plants for photosynthesis. You guys do not understand how important this page is. Very, very, very important. Okay. Bye. What's after is that we're going to be talking about the biochemical tests for the carbohydrates. We have the Benedict reagent, the non-reducing, and also the iodine test. Okay. The Benedict reagent can be used to test for the presence of reducing sugar. All monosaccharides are some disaccharides, for example maltose, are reducing sugars. These are therefore sugars that can donate an electron to the Benedict reagent. Benedict reagent is an alkaline solution of a copper 2 sulfate. When a reducing sugar is added to this and heated, it forms an insoluble red precipitate copper 1 oxide. The Benedict test is summarized below. You first add 2 cm cube of the food sample to be tested. It needs to be in liquid form to begin with, and then add 2 cm cube of Benedict reagent. Now heat the mixture gently in a water bath for 5 minutes. If the solution turns brick red, then a reducing sugar is present and it's a positive result. If it does not and stays the same, then it's negative result. Okay. Although some disaccharides are reducing sugars, other disaccharides as well as polysaccharides are non-reducing sugars, and therefore the Benedict test has to be altered in order to test for these. The process occurs as follows. First of all, you add 2 cm cube of food sample and must be in a liquid form as well, and is added to 2 cm cube of Benedict reagent. Then it's placed in a water bath for 5 minutes, gently warm. Exactly the same as the reducing sugar. But here, if the color does not change from blue to brick red, then a reducing sugar is not present. If you want to look for a non-reducing sugar, you add another 2 cm cube of the same food sample is then taken, and then 2 cm cube of dilute hydrochloric acid of HCl is added. The test tube is then placed in a water bath for 5 minutes. The dilute HCl will hydrolyze the disaccharides and polysaccharides into their constituent monosaccharides. After this, some sodium hydrogen carbonate is added in order to neutralize the test tube, as the Benedict reagent will not work in acidic conditions. The pH paper is used to check the solution is neutralized. The solution can now be retested by adding 2 cm cube of Benedict reagent to the solution and placing in a water bath for another 5 minutes. If a non-reducing sugar is present in the original sample, then a color change from the blue Benedict reagent to brick red will be observed. And then lastly, the iodine test, which is the test for starch. If the solution turns blue-black in color from orange-brown, then starch is present. Now we go to the lipids, where it has the emulsion test as its chemical test. Lipids are biological molecules made of carbon, hydrogen, and oxygen, which are only soluble in organic solvents such as alcohols. For example, if you put oil in water, it's insoluble. The main lipid types are triglycerides and phospholipids. Triglyceride, which is one of the main types, is that triglycerides are lipids made of one molecule of glycerol and three fatty acids, as you see in the diagram, joined by ester bonds formed in condensation reaction. So all biological molecules are formed by condensation reaction. However, the bonds change. In carbohydrates, it's glycosidic bond. In triglycerides, in lipids, it's ester bonds. These are many different types of fatty acids, over 70 different types, which vary in chain length, presence, and number of double bonds. The presence of double bonds changes the name of the lipid. They can either be saturated or unsaturated. These are explained below. The saturated basically has the word S, which says it's solid and single. Saturated solid means it's solid at room temperature, because you're gonna find MCQ questions about that, even though in the Excel, etc. And single means it has single bond. Okay, so the saturated lipids, such as those found in animal fats, saturated lipids do not contain any carbon double bond carbon. It's only carbon single bond carbon and one carbon double bond O. The unsaturated lipids, U is a vowel like oil, which says it's like liquid, and like olive oil, and as it's present in plants, and it's double bond. So the unsaturated lipids, which can be found in plants, unsaturated lipids contains carbon-carbon double bond. Yeah, and unsaturated, I said S is for solid, as solid is like butter in animals. Double bond, and the presence of a double bond means that the molecule is able to bend. As a result, unsaturated fats cannot pack together as tightly and are therefore liquid at room temperature. So the unsaturated is liquid, like Q, olive oil, as it's in plants, and as well saturated is single, so single bond. Saturated is solid, so solid is like butter as animal. Okay, the triglycerides structure related to their properties. First of all, we have high ratio of energy storing carbon-hydrogen bonds to carbon atom, and therefore they are excellent energy store. Okay. Second point is that the low mass to energy ratio mean that they are a good storage molecule with a lot of energy being stored in a small volume. This is beneficial for animals as it's less mass to move around. Third, being large and nonpolar lipids are insoluble in water, and therefore their storage does not affect the water potential of cells. Lastly, a high ratio of hydrogen oxygen atoms means that triglycerides release water when they are oxidized, and therefore provide an important source of water for organisms to live in dry environments. Phospholipids. In phospholipids, one of the fatty acids of a triglyceride is substituted by a phosphate containing group. Phosphate heads are hydrophilic, which means they love water, and the tails are hydrophobic, has phobia from water, hates water, and as a result, phospholipids form micelles when they are in contact with water. The molecule is therefore known as polar. The structure of phospholipid is shown below. As you see over here, phosphate CH2 CH COO CH2 COO. Okay. The phospholipid structure related to their properties. Basically, in an aqueous environment, being polar means a bilayer can be formed, and the hydrophilic heads of the phospholipid can be used to hold at the surface of the cell surface membrane. Their structure allows them to form glycolipids with carbohydrates, which are important on the cell surface membrane for cell recognition. And now we go to the emulsion test. Emulsion test can be used to test for the presence of lipids. This is carried out as follows. You first take a completely grease-free test tube and add 2 centimeter cube of the sample to be tested and 5 centimeter cubic of ethanol. Shake the test tube thoroughly to dissolve all the lipid in the solution. Add 5 centimeter cube of water, and as you know, it's soluble in alcohol or like organic solvents, not in water. And then shake in organic solvents, as you see over here. Then you add the water and shake gently. A cloudy white color indicates the presence of lipid. As a control, repeat the experiment using water as the sample, and the final solution should remain clear. Okay. Now we go to another biological molecule called proteins. What do you know about proteins, and what do you want to know about proteins? We know that amino acids are the monomers from which proteins are made, and that and that we need to know that amino acid contain an amine group NH2, carboxylic acid group COOH, and the variable R group, which is a carbon-containing chain. The basic structure of an amino acid is shown, as you see over here: NH2, CRH, C double bond OOH. And there are 20 different amino acids, each determined by their different R groups. Amino acids are joined by peptide bonds, so they all are joined by condensation reaction. Here in the amino acids, they are joined by peptide bonds. In this reaction, a molecule of water is formed with the process shown in the image below, as you see over here. A dipeptide contains 2 amino acids, and polypeptide contains 3 or more amino acids. The structure of the protein are divided into 4: primary structure, secondary structure, tertiary structure, and then the quaternary structure. The structure of proteins is determined by the order and number of amino acids bonding present and the shape of the protein. The primary structure of a protein is the order and number of amino acids in a protein. This primary structure contains the initial sequence of amino acids and will therefore determine the protein's function in the end. The secondary structure is the shape that the chain of amino acid makes, either alpha helix or beta plated sheets, and the hydrogen in the NH has a slight positive charge whilst the oxygen in the CO has a slight negative charge, and as a result, weak hydrogen bonds can form, leading to alpha helixes or beta plated sheets. Again, the primary structure of a protein is the order and number of amino acids in a protein. This primary structure contains the initial sequence of amino acids and will therefore determine the protein's function in the end. The secondary structure is the shape that the chain of amino acids makes, either alpha helix or beta plated sheet. The hydrogen in the NH has a slight positive charge, whilst the oxygen in the C double bond O has a slight negative charge, and as a result, weak hydrogen bonds can form, leading to alpha helixes or beta plated sheets. Now we go to the tertiary structure. Tertiary structure of proteins is the 3D shape of the protein and is formed from further twisting and folding. A number of different bonds maintain the structure, and these are the disulfide bridges, ionic bonds, and hydrogen bonds. Again, the tertiary structure of a protein is the 3D shape of the protein and is formed from further twisting and folding. A number of different bonds maintain the structure. These are the disulfide bridges, which interactions between the sulfur and the R group of the amino acid cysteine. These are strong and not easily broken. The ionic bonds formed between the carboxyl and amino groups that are not involved in the peptide bonds. They are easily broken by pH and are weaker than disulfide bridges. The hydrogen bonds are numerous and easily broken. Proteins can be globular or fibrous. Globular proteins such as enzymes are compact, whereas fibrous proteins such as keratin are long and thus can be used to form fibers. I want to also tell you something about the quaternary structure of the protein. Okay, that you need to know, that the arrangements, they have also arrangements of multiple polypeptide chains or subunits in the protein complex. So basically, the quaternary structure is basically that the protein consists of more than one amino acid and chain. That's it. Okay. Then we have a good part for the fibrous and globular. That the globular and the fibrous, basically the fibrous, as we said over here, such as keratin, are long, and the globular has globular shape, and which is basically spherical, while the fibrous protein have an elongated or fibrous shape. As we said, the globular protein are typically water soluble and perform a wide variety of functions in the body, such as enzymes, transport proteins, and hormones. So yeah, and the globular proteins are water soluble, as only the hydrophilic components of the protein are in the outer surface, and the globular proteins are soluble in water. What's left? That's it, right? Okay. Yeah, and you need to also know that example of the globular protein is the hemoglobin, which is made up of 4 globular subunits. Hemoglobin is a quaternary protein made up of 4 tertiary globular subunits. 2 of these subunits consist of alpha chains, while the other 2 chains beta chains. Each of these globular subunits is linked to them. It is linked to heme. Each globular unit is covalently bonded to heme. Heme is not a protein, therefore it's called a prosthetic group. Heme contains iron, which oxygen binds to. Hemoglobin is considered a conjugated protein, as hemoglobin is a protein that is associated with non-protein structure. It's called a conjugated protein. Fibrous protein, as we said, are long chains, and they are made up of repeated amino acid sequences that form long polypeptide chains. These chains twist together to form fibrous proteins, and fibrous proteins are water insoluble. Example of the fibrous protein which is important is the collagen. Collagen is a strong protein due to the types of bonds in a structure. The proteins of collagen are joined together